Okay, hi everyone. Happy Friday. Um, let's continue talking about B cells and how B cells finish their development in the bone marrow and what happens next once a B cell has made its immunoglobulin on its surface and what sort of selection processes it has to undergo before it's really allowed to go to work as a functional part of our immune system. And in addition, we'll kind of get a little jump start today on also talking about the development of T cells. And that discussion we'll continue on with in next week's lectures. So remember, this is where we kind of left off last time. We had been talking about the development of B cells that take place as B cells are first born, just like any other cell of our immune system and any other cell that's part of our blood, right? All of those poetic cells come from stem cells in our bone marrow. And importantly, we're talking about the red bone marrow, or the bone marrow that's part of our long bones, like our arm bones, our leg bones, and also in our pelvis. So that's what's represented in yellow in this cartoon that we had looked at. And we said that that's where B cells are first born. They have to go through the whole process of making VDJ recombination, getting it to work, right, which is itself not a very simple process. It's very complicated. About half the B cells can't even make something like the heavy chain of their immunoglobulin. So right off the bat, you lose like half of the B cells that are developing in the bone marrow. And then those who do make a heavy chain, then they have to make a light chain, right? And so, most of those probably can make a light chain because we have two different opportunities to make a light chain, right? We have both the kappa and the lambda loci in our chromosomes that have all the gene fragments to make the light chain. And then the cells that have immunoglobulin on their surface, we still consider those immature, right? Because we said that at that point, the sort of molecular wiring of the immunoglobulin and the way it communicates with the nucleus of the B cell is, in a sense, backwards at that point because of the important consideration that the shape of the antigen binding domains of these immunoglobulins, because they came from VDJ recombination, right, are very randomly decided. Remember, that's the whole point of VDJ recombination, is so that you can make hundreds of millions of different immunoglobulin antigen binding domain shapes every single day. And last time we ended talking about the fact that, yeah, if you're going to be making these shapes, these antigen binding shapes randomly, there's a really good chance that you'll end up with B cells that have immunoglobulins on them that end up recognizing and binding to perfectly normal shapes that are inside of your perfectly normal tissues and cells, right? So that's why B cells have to undergo a screening process to make sure that they are not autoreactive. So all of that takes place in the bone marrow. And today we'll finish talking about that selection process and look at what happens as the B cells then shift to places where they go to work. In other words, secondary lymphoid tissues, places like the lymph nodes, spleen, and specialized tissues around our intestines, the Peyer's patches. All right. So let's review when we talked about this whole screening process. We said that the brand new immature B cells that have their IgM on their surface, remember IgM is the first isotype of immunoglobulins that B cells make, right? And these are still stuck to the surface of the B cell as B cell receptors. We said if you have immature B cells in the bone marrow that have an opportunity to now see if they bind any sorts of signals that are in the bone marrow, for example, perfectly normal antigens that are on the surface of like this bone marrow cell here, right? If that B cell did not use up all of its V and J blocks to make its light chains, then, the B cell has an opportunity to try again. The oven mitt, the RAG protein complex goes to work again and tries to choose a different combination of V and J so that the light chain part of the Ig maybe is slightly different now, but maybe that slightly different shape is enough so that this B cell stops autoreacting against the gray triangles that are being presented by your normal body's healthy bone marrow cell. Right? If the B cell can't succeed with that, either because it doesn't have any more VJ blocks because it used them all up in the first place to make its IgM, or it keeps trying to do new ones, but after a couple times it just can't do it, then this B cell is just told, sorry, you know what, we can't let you loose inside of the body because obviously you're going to cause trouble if you're auto-reacting because your Ig has a shape that recognizes self-antigen. So that B cell, right, just undergoes apoptosis. It's just told, die, take yourself out of the picture here, right? So if the B cell, though, either did not have autoreactive properties with stuff in the bone marrow to start with, or, like the one on the left here, it got a chance to go back and it changed its light chain a little bit, and now its IgM no longer reacts to things that are in the bone marrow, right? Then it can be released into the blood. So up until now, this tolerance screening, that's what we call it, right? How well does the brand new B cell tolerate self signals, right? That so far has been in the bone marrow. So we call this central tolerance training. It takes place in the bone marrow. It's the first kind of tolerance training that the cell undergoes. After it succeeds in passing central tolerance training and is released into the blood, oh boy, right? If you thought that there were a lot of self-antigens in the bone marrow, now this B cell is going through the circulatory system. It's encountering so many more examples of three-dimensional shaped molecules that are floating around in the blood with it, right? So at this point, it's still critical to realize this B cell is not ready for action yet, right? It's still undergoing this kind of tolerance evaluation. But now in the blood, we call this peripheral training. So now it has an opportunity to go through the blood, right, and see if it unfortunately binds to something in the blood. Here you see this B cell, unfortunately, even though its IgM did not bind anything when it was still in the bone marrow, right? Once it's gone into the blood and it's going around, oh, it binds these little red diamonds, right? And these red diamonds are meant to represent some sort of molecule that's very commonplace or normal in your blood, right? That comes from your own tissues. So clearly we have a problem, right? So unfortunately, because this B cell has left the bone marrow, it's no longer in that correct tissue environment to try to do anything else with like VDJ recombination or try again or whatever. So at that point, right, if its IgM starts binding up self-antigen, this B cell, unfortunately, right, is not going to go any further. So what happens is the B cell begins to express a lot more IgD on its surface. You'll recall that IgM and IgD, right, two of the 5 subclasses of immunoglobulins, they only differ by the different stem parts, right? The little red arm parts that are normally, well, on the other cartoons, they show the antigen binding domains as red on the Y-shaped immunoglobulin, right? And then the stem part was like navy blue before. The antigen binding domain stays the same. It's just now the cell, through alternate mRNA splicing, starts making a lot more of the IgD form, right? And so this IgD form will also bind that red self-antigen, and that will send a strong signal into this B cell that, you know what, no, you're not going to work, right? You don't have to die right away, but you're just not going to go any further in your B cell development. So this B cell then undergoes a process that we call, it becomes anergic. Anergic means basically inactive or shut down, right? It's just kind of there, and eventually it's just going to end up being recycled. All right, so that's important to think about, right? That once the mature B cell, once the immature B cell is formed in the bone marrow, it has to go through these critical tolerance training stages to make sure that it is not going to auto-react with, the things that are normally in your body, right? So if that happens, eventually so if a B cell passes both central tolerance training and peripheral training, right, in the bone marrow and then in the blood, eventually it can make its way to secondary lymphoid tissues like the spleen or the lymph node. Right? And that is where the B cell will finally finish maturing and being ready to go to work. So let's talk about these secondary lymphoid tissues, right? Lymph, what is lymph? Well, lymph refers to components of our lymphatic system. I think a lot of times, students, you're well aware we have a circulatory system, right? And the circulatory system has things like arteries, veins, capillaries, whatever. I think a lot of times students are vaguely aware, yeah, you have this other like lymphatic system, right? Lymph nodes and things like that. But if you're ever asked, well, how is that set up compared to our circulatory system? I think most people would just sort of assume, yeah, they're somehow like connected or something like that, right? When in reality, the truth is our lymphatic system and our circulatory system are kept very separated on purpose, right? They work together, and in many ways they overlap physically, but they are really only a couple of very specific locations in your body where you have a connection taking place between the two. This cartoon kind of illustrates in a super simplified way how our lymphatic system relates to our circulatory system, right? So our circulatory system, you can think the main part of it is like our heart, right? If you're going to discuss the circulation, you think about starting with the heart. And of course, the heart pumps blood to the lungs, and the lungs get the blood oxygenated, and the oxygenated blood comes back to the heart, and then the heart pumps it out to circulate around the body, right? Things like out of our aorta, it's going to enter our atrial system, our system of arteries that branch and become progressively smaller and thinner vessels carrying the blood until you reach the level of different tissues where the teeny tiniest, thinnest kind of forks in the road of these arteries are down at the level of capillaries, right? So in our tissues, we have capillary beds. These are where the vessels are so thin that literally the wall of your capillary is only one cell thick, right? And that's designed that way because things that are being carried in the blood, like nutrients, gases like oxygen, need to be able to just diffuse out of the capillaries into the surrounding tissues where these things are needed. Right? And at the same time, waste products from the tissue, like carbon dioxide, like other metabolites, waste products that need to be removed from the tissue, they diffuse into the capillaries. Right? So that's what's going on here. And then that collected blood, right, the spent blood, if you want to call it that way, we typically represent that now as like blue, right? I don't know why we've traditionally called like oxygenated blood red, deoxygenated blood blue. I mean, I think there is some evidence that oxygenated blood is redder in appearance, right? But the hemoglobin that's in charge of carrying around things like oxygen is the same hemoglobin that then, once it gives up the oxygen, happily then picks up CO2 and carries the CO2 back through progressively bigger and bigger vessels, right? From little venules to veins, and finally up to the size of the biggest veins like the vena cava that feed back into the heart. Let's appreciate here how the lymphatic system is kind of drawn here as being kind of like draped over the circulatory system. Let's zoom in on the capillary bed here. What you'll see is the lymph vessels represented in green. Yeah, these are separate vessels, okay? And look how they are intertwined with the capillaries. But importantly, you never see an example in this illustration where a green lymphatic vessel is directly plugged into or connecting to a capillary, right? That means what? The blood never actually runs from your circulatory system directly into your lymphatic system. What is happening here is all these thin little lymphatic vessels have open ends, right? So these are like tubes that have open ends, and what they're doing is they are draining all the stuff from the surrounding tissue. So as you have water and nutrients coming out of these capillaries to feed the tissues, right? Then there's going to be a lot of stuff, things like extra water that you need to now get rid of. Otherwise, if your tissues kept filling up with water, what would happen? Your tissues would get really bloated with water. And when that happens, what do we call that? Right? We call that edema. So the swelling of different areas, like around your ankles and things like that, right, is because the fluid is not being drained properly from the tissues. So it's kind of like gutters, right? Gutters that are like on a roadside. Stuff is just going to drain into the ends of these lymphatic vessels, and they will be carried away. Where will they be carried? You can see back up here, they'll be carried through these different specialized tissues known as lymph nodes. Right? And these lymph nodes that we are going to spend a lot of time understanding what they do and why they're so critical for our immune system. Right? These lymph nodes essentially clean up the dirty fluid. And once that fluid has gone through a whole series of lymph nodes, because these lymph nodes are all interconnected, kind of feeding each other, eventually the cleaned up fluid, which is mostly just water at that point, is introduced back into the blood, right up here. Right? So this is just one of two places at the level of our shoulders where the lymphatic system empties the cleaned up fluid to rejoin the blood, right? Because remember, your body needs to maintain its water, right? So once the water has left your circulatory system and is in your tissues, and the lymph vessels sort of drain that so that your tissues don't swell up, what are you going to do with that water? Right? You say, well, why doesn't it just go to your kidneys and you can like pee it out? Yeah, but what a waste, right? Then you all day long you'd have to keep drinking water, right? So you'd rather clean it up and save that water and get it back into your blood, right? So that happens at the level of our shoulders, at the lymphatic duct and thoracic duct on either side that enters the major veins that feed back into the heart. So right before the heart, this cleaned up fluid is reintroduced. I also wanted you to notice that in a cross section of these lymphatic vessels, you'll see that there are little valves, right? Little valves that encourage the fluid to go one direction, right? In this case, in the illustration, the little arrows are showing you that the lymph fluid is going up, right? Up against gravity through your body. So I mentioned it's like a system of gutters. So think about this, right? So it's like, here's your neighborhood and here's your neighbor who's very wasteful, running the sprinkler. Actually, this is probably not our neighborhood. I don't know what this is. I think it's a chunk of ice here, right? So this is probably somewhere on the mainland right now. Although on the mainland, I think most parts are covered in like 2 feet of snow right now. So we're lucky, right? We're lucky here in Hawaii. Anyhow, so the water that comes out of the lawn sprinklers is fresh water, right? It's not dirty water. It's fresh water that came into the property and now is coming out to sprinkle the lawn. So that's like things like the fresh water that diffuses out of the capillaries, right? That's carrying with it things like nutrients and oxygen, things like that. So think about the fresh water by the time it's collected and is running through the gutters on the side of the road, right? It's kind of gross. It's collected all kinds of waste and debris, right? Stuff on the road. Maybe there's dead bugs from the grass, bits of grass itself, dirt. And then it all drains into this sewage intake so that it can go off somewhere and get cleaned up, right? Importantly, in this illustration, does this sewage water that's connected simply get fed back to the lawn sprinklers again? No, right? The fresh water and the wastewater is always kept separate. And so that's just like the way our lymph system and our circulatory system are set up, right? Our blood and our lymph fluid, right, never mix directly as part of it. So here's sort of an anatomical overview of our lymphatic system. So I mentioned that along the way through our lymph system, we have these special regions of tissues known as lymph nodes. I think you guys are all sort of familiar with lymph nodes, right? Like you've heard about when you have infections or something like that, you might get swollen lymph nodes, right? Your nurse or doctor can feel around your throat, right? Or under armpits. So sure enough, right, that's where we have the greatest concentration of lymph nodes. Lymph nodes are not spread out evenly or uniformly throughout the body. You can see they are collected in regions like the neck or the armpits or around the pelvic region. Right? So these lymph nodes then each represent a separate spot where the lymph fluid can be cleaned up a little bit before it gets passed onto other lymph nodes. Right? And the lymph nodes, because they're kind of random in terms of how they are interconnected within these different regions here, right? It means that if you were to follow some lymph fluid that's coming from the tissue of the leg and it's draining and coming up into the lymph nodes here, right? You wouldn't necessarily know or be able to tell what path through these different lymph nodes it'll take. But the point is, because it's passing through different lymph nodes, eventually by the time the fluid gets up to the level of our shoulders, it's all cleaned up, right? And that water can be reintroduced into our circulatory system by joining up with our major veins that are at the level of the shoulders. Okay, so when you are familiar with lymph nodes, because you say, yeah, I know that during certain types of infections, they get swollen, right? What's that about, right? Right, that should give you a clue. Lymph nodes are very important in the functioning of our adaptive immune system. So let's take a look here. So a lymph node is kind of like a kidney-shaped or a bean-shaped tissue structure, right? It's pretty well defined because it has a capsule around it in which you have these different regions of tissue. Right? Now, we mentioned that these lymph nodes would be connected to our lymphatic system. So that means you should imagine certain lymph vessels are connected coming in, certain lymph vessels are connected going out. Right? So up here, that's what's shown here. This little entry point here and this entry point here would represent what we call afferent lymphatic vessels. In other words, stuff is coming in through these regions that might be coming from the tissue that was being drained, or maybe from another lymph node that was upstream of this. Right? In terms of the way out, there's really only one way out of this lymph node, and that is through the efferent lymphatic vessel, this big one here. All right, so that means the lymph fluid is flowing into this kind of bean-shaped structure through multiple afferent entry points. Right? Again, right, because these could be coming from a different lymph node or from tissue, different places are draining into this particular lymph node, but that lymph fluid, once it's in here and has an opportunity to be kind of examined by our immune system, maybe cleaned up a little bit by cells, right, it will leave through the efferent lymphatic vessel to go on to the next step. And the whole point is, hopefully you can figure this out, right? This efferent lymphatic vessel, as it goes off the screen here, becomes the afferent vessel leading into the next lymph node. All right. So that's the lymph fluid. Here you see there are blood vessels are also attached to it, right? And you're like, wait, I thought the blood and the lymph don't mix, right? Yes, that's true. But remember, because this is living tissue, right? Your lymph vessel is living tissue full of living cells. It needs to have the blood delivered to it also, so it can get oxygen and nutrients and wastes can go away also, right? So here you see blood vessels arriving at the lymph node. The way the cartoon's drawn is kind of weird because imagine these dark blue purple vessels are actually coming up behind the lymph node in this cartoon, and then they're entering the lymph node from behind over here. And here you see that purple blood vessel cut in a cross section, right? Because it's coming through the screen at you. As a result, blood cells, like red blood cells, things like that, they'll pass through here, come up behind, pass through the tissue, right? And nutrients will diffuse out of the blood vessel to feed the tissues of the lymph node. But the blood cells themselves, like red blood cells, never enter the lymph vessel. They just pass through, or the lymph node, they pass through, and then eventually continue on through the blood to the next, tissue destination or whatever. But among the cells that are coming through the blood are things like our B cell, right? Our B cell that has managed to pass central tolerance training in the bone marrow, peripheral training in the blood, right? And now, as part of going around in the blood and not getting into any trouble, eventually, just through chance, it's going left, right at each fork of like an artery or whatever. Eventually, that B cell makes its way to this lymph node. So it comes up behind it in the blood, and this region of the blood vessel that's drawn here as this kind of like, I don't know, purple donut, right, represents a specific region of the blood vessel known as a high endothelial venule, or HEV. The cells that make up the wall of this HEV have special signaling molecules, right, special receptor-like things, where the B cell, as it passes through, right, interact with those, and that's how the B cell knows, hey, I've been going through the blood, now I'm passing through a lymph node, because the wall of the blood vessels has these things that now I can attach to, and the B cell will squeeze its way out of the blood and enter the tissue of the lymph node. Right? Inside the lymph node, the B cell will have an opportunity now to interact with other lymph node cells, right? And importantly, what? Potential antigens that were brought in here through the lymph fluid that was draining from tissue, for example, from the site of infection. Right? That B cell then may or may not have the opportunity to activate right in here, but eventually the only way out of this lymph node is out through the efferent lymphatic vessel, right? Because at this point, right at this point here, this very first red arrow, the B cell has left the blood circulation, right? It's not going to re-enter the blood circulation. It is now part of the lymph system, right? So it's riding all the lymph fluid, eventually going up also and being reintroduced into the blood at the level of your shoulders, where this whole cycle begins again. It goes through the blood, finds its way to a lymph node, exits into the lymph node, may or may not find anything interesting to react with, and it just goes around and around between the blood and the lymph. Importantly, the first time a B cell makes it to a lymph node, that's a big event, right? Because up until now, you're like, yeah, the B cell's good because its immunoglobulin didn't bind anything, so it didn't get into trouble, right? And importantly, the fact that it can leave the high endothelial venule in the lymph node and actually go into the lymph tissue, that's the other critical thing that this B cell has now shown it can do, right? Because it means it has the right receptors on its surface to recognize that it's in the HEV vessel, and so it was able to leave, right? So in this cartoon here, the light blue in these panels is the tissue of the lymph node, right? Surrounding a little capillary that's running through it, and this is that special HEV region where the cells on the wall have those signals that the B cell that's passing through gets stopped. And then the B cell you see in panel 2 over here squeezes out, right, and is now in the lymph tissue. In the lymph nodes tissue, there are dendritic cells, like these yellow ones here, and these other types of stromal cells that are part of the tissue, right? So these dendritic cells reward the B cell by encouraging the B cells to travel to a part of the lymph node that we call the primary follicle. Okay, let me back up a moment because I should have illustrated. These different regions of the lymph nodes tissues each serve different functions. Right? What we are talking about right now are these yellow regions, which are called the primary lymphoid follicles. So this is where brand new B cells that come into the lymph node are encouraged to go to get their reward, right? The reward for making it this far. So here in the primary follicle, this big blue cell is a special dendritic cell, which we call a follicular dendritic cell, right? So it is a dendritic cell, right? But it's a very special type of dendritic cell. It hangs out in those primary follicle regions. And when the brand new immature B cells arrive, the follicular dendritic cell here rewards those B cells, right? By basically fixing their wiring now of their immunoglobulins, right? Because up until this point, remember we said functionally their immunoglobulins are kind of wired up backwards, right? At this point, if anything bound to them, the B cell would actually go anergic and shut down because the overwhelming chances are that whatever it's bound to in the blood is actually self-antigen, right? You might say, yeah, but what if it actually was successful in binding, some sort of foreign antigen? Isn't that a shame if that cell gets lost? Yeah, it is, but it's a cost benefit kind of thing. It's far more likely that an immature B cell, if it binds something in your blood, right, it's a self signal that it's binding to. So Mother Nature evolutionarily plays it safe, right? Up until this point, if anything bound to that B cell is just taken out of the picture. But if the B cell made it through the blood and made it into a lymph node, right, its first visit to a secondary lymphoid tissue, it is rewarded, right? And so there are different types of signals that then rewire the IgM now. So now, if anything binds to the IgM, instead of it being inhibitory, right, it's going to be stimulatory. And that B cell is now considered mature, right? It is ready to go to work. It is ready to be released now to go through its life, going between the blood, the lymph, the blood, and the lymph. And if it ever ends up binding something from this point on, wow, then you're in business, right? So this last panel just reminds you of that B cell is released into the lymph system. Eventually, it joins back up into the circulatory system, goes through your blood again, only to eventually make it back somewhere to maybe a different lymph node, right? I like the highly accurate human anatomy drawing here, right? Okay, but it gets the point across. All right, so let's break this down in a little bit more detail, right? So that you can appreciate what's going on. So here we have a B cell that is mature, all right? So in other words, it made it to a lymph node somewhere and it was rewarded by having its IgMs kind of like rewired. So now that they'll work to send it a positive activating signal. And then that mature B cell was then allowed to go back into circulation. That B cell is still considered naive, right? Naive means that to this moment, its IgMs have not bound to anything yet, right? So chances are, if this is one out of several hundred millions of B cells that your body, cooked up today, right? Chances are it will never end up binding to anything, right? But it's circulating around, and let's say now it makes its way to another lymph node. So it was going through the blood, comes up behind this lymph node, comes through the high endothelial venule area, right? Which releases it into this blue region, which we call the T cell area. This is where T cells hang out inside the lymph node. Right? So step one is the naive B cell enters the lymph node. Ooh, look what's happening here. This particular lymph node that this B cell entered by chance, it's receiving all kinds of goodies through the lymph that's flowing in through the afferent lymphatic vesicles, right, that are feeding into this lymph node, including, what do we have here? Dendritic cells that are covered with maybe some sort of antigen from a pathogen, maybe the pathogen itself, like this bacteria, because maybe off the screen here to the upper left, right, maybe this lymph node is the closest lymph node to maybe a place where you have a cut on your arm. So a lot of the antigens that are being drained by the lymph system in the tissue here, right? In that sewage wastewater is all kinds of evidence that there is an infection going on. Bits and pieces of antigen that are being carried by things like dendritic cells, bits and pieces that are just floating, or maybe complete bacteria itself are all coming in here. And guess what? This is this B cell's lucky day, right? Because it just so happens that its immunoglobulins are the right shape to bind the little red dots here, right? So what's going to happen then is the B cell will activate, and the B cell, it'll find a partner, because these T cells, some of these T cells are helper T cells that have T cell receptor that by chance are recognizing certain peptide fragments, right, that are draining in here as evidence of infection also. And then the T cell will pair up with the B cell. So step two is you'll have T cells that help tell the B cell what to do. Remember, right, when we studied T cell activation, we said that one scenario is a B cell that is binding stuff like antigen on its immunoglobulins, and the B cell is taking these antigens in as part of just recycling these immunoglobulins on its surface. So as a result, the B cell is presenting little bits of peptide, right, on its surface on MHC2, right? And in this cartoon, this helper T cell happens to have a T cell receptor, right, that recognizes that red dot, the peptide that this B cell is presenting it. Okay. Then the T cell needs to tell the B cell what to do, right? It's a helper T cell. Right? In many ways, the B cell is completely clueless at this point, right? its immunoglobulins have bound things. So it's like, whoa, right? I'm psyched. My immunoglobulins are working, right? But it doesn't know what to do next. So that's why the helper T cell needs to tell the B cell what to do. And remember, it does that by the helper T cell releasing cytokine messages. Right? So it encourages the B cell to start multiplying, making perfect clones of itself. Why? Because think about it. If this immunoglobulin that's on the surface of this B cell suddenly is valuable, right? Because up until now, chances are you thought, hey, I don't think this immunoglobulin is going to amount to anything, right? Like most immunoglobulins don't amount to anything. But look, it did. It bound to the antigen. This B cell has the unique recipe for that immunoglobulin, right? This B cell has suddenly become very valuable. You don't want to lose this B cell. So right away, the T cell tells that B cell, make many copies of yourself, right? So that's what you see here. Some of these copies are told by these helper T cells, okay, your immunoglobulin is the best thing we have right now that recognizes this antigen. Therefore, some of you go ahead and become plasma cells, because what we want you to do now is start cranking out buckets and buckets of the immunoglobulin as antibodies, right? So some of these B cell clones are encouraged to go ahead and become plasma cells. And so they become factories of making antibodies, right? And now these antibodies just start flooding your circulatory system. And some of that, right, will eventually make it back to the site of infection, where these antibodies will now start binding to the bacteria, right? Some of these other B cell clones are encouraged by the T cells to go up into the primary follicle area again, where they start forming these special clusters of B cells known as germinal centers. And what do you think is happening in these germinal centers? This is where hypermutation of the immunoglobulins is taking place, right? So these B cells are now dividing and doing the random kind of mutations of their immunoglobulin locus to see if they can come up with more refined, better fitting shapes to actually bind the bacteria, right? So step 3, in the germinal centers, B cells hypermutate to refine their Ig affinity. And that means that as you get better and better B cells up here, some of them come back out and the T cells tell them, ooh, right, your Ig is better than the last group of Igs that those B cells made. Why don't you turn into plasma cells now, right? So as a result, throughout an infection, right, as your adaptive immune response is doing this, your lymph nodes send out wave after wave of better and better antibodies, right? And each wave, the quality of the antibodies is better than the last wave, right? Lastly, after the infection is beaten by your adaptive immune system, right, you don't want to have to go through this whole process again, because what are you feeling while this is going on right now? You're feeling lousy, right? You've gotten really sick, because this is like a week after you first got infected with this, and it's taken that much time for this whole selection process to take place and to really start building up appreciable numbers of selected B cells, right? So you're feeling miserable, right? You have a fever, you've been throwing up. I mean, the truth is, right, I always remind students of this, when you're sick and laid up in bed and just binge watching Netflix or something, right? I mean, you're dying, right? Because the pathogen is taking over your body, and the pathogen is not going to stop, right? Just because it gets bored of you or it feels bad, right? It's going to keep going until there's no more resources for that bacteria to use. And that means when you're dead, right? So if you survive that by being able to select B cells and produce better and better quality antibodies that ultimately opsonize the bacteria, right, way better than what your poor innate immune system tried to do but couldn't, and that's what eventually saves you, right, because those antibodies were able to do things like attract complement or get other immune cells to pay attention. You don't want to have to start from scratch again next time that you end up with bad clown coming into your body again, right? So some of the highly refined B cells that are up in these germinal centers, once the infection is over, these B cells represent the B cells with the best cookie recipes that they came up with, right, through this infection. Those B cells are super valuable, right? So some of those stick around after the infection, so that if you do, weeks later, months later, or maybe even years later, have bad clown come back, right? Those B cells already make the best quality IG on their surface that you came up with last time, right? So they represent the starting point of your defenses the next time around, right? And because they already have those really good immunoglobulins, when they're told to turn into plasma cells and start making the first wave of antibodies, those antibodies will be really way better than the first wave of antibodies you made the first time you were infected with bad clown. To date, we don't know how long these memory B cells stick around, right? It was thought that individual B cells, it's not that an individual B cell stays in your body for years. It's most likely that over time, an individual memory B cell might be told to divide, right? And maybe one of the daughter cells dies, but the other continues on. But now we actually have some evidence that certain plasma cells, right, can live for potentially many years, right? And that's something that we didn't really appreciate before, but just in the last half year or so, there's been some new findings that suggest that we have long-term memory plasma cells that stick around. Okay, so that kind of wraps up our discussion about the B cells and how they mature, undergo tolerance training, and then ultimately are put to work, right? Obviously, as our semester goes on, we'll talk more and more about B cells and T cells working together. But for now, let's shift our focus onto looking at how T cells develop and what they go through, right? So remember, a big distinction is that while B cells and T cells both are born in the primary tissue, right? In this case, the yellow boundary means inside of your primary, like tissue, for example, your bone marrow, right? The B cells, as we finished talking about, by the time they leave the bone marrow, they already have IG on their surface, right? Because they've already not only made IG successfully, but they've undergone central tolerance training in the bone marrow to make sure their version of the IG doesn't get them into trouble. So when the B cell leaves the bone marrow, right, it already has IG on its surface, and then it makes its way to secondary lymphoid tissues like lymph nodes, where they then ultimately hope, right, if they're so lucky, to be selected to become a plasma cell. So T cells are a bit different. So T cells, as they are developing in the bone marrow, by the time they leave the bone marrow, right, they still don't have anything resembling T cell receptor on their surface, because this kind of purple cell in the cartoon, right, when this thing leaves the bone marrow, this thing could become a natural killer cell or a T cell. In order for it to become a T cell, it has to make a little detour, a little pit stop in a place that we call the thymus, right? So the thymus is here. It's located in front of the heart between the lungs, and it's in the thymus. That's where these cells are encouraged to become T cells. In other words, that's where they first start doing their VDJ recombination to kind of come up with a T cell receptor. Right? So here's the thymus. Notice that in an adult, the thymus is quite a bit smaller proportionately than an infant, right? And so this is really important because this has to do with how Mother Nature designed us with what to expect. Right? As an infant, right, you're born without much of an adaptive immune system at all, right? So the moment you're born, oh boy, you are like a sitting duck for all types of nasty pathogens, things like bad clown, right? So that's why infants have a very large thymus, because what it's trying to do is to get up and running, right? Try to start making some T cells, and that's a huge priority. As we get older, though, the thymus shrinks. Right? And you might say, well, why is that? Why is that? Because it's not like in our life we stop needing T cells, right? But Mother Nature never intended us to live as long as we do these days. Okay. And so as a result, the thymus is only kind of designed to hang out for a couple or maybe 3 decades of full activity before it begins to kind of lose its function. Right? Here is a cross section of the thymus. You might know the thymus by another name, in cooking, like fancy gourmet cooking. This is called sweetbreads, right? From like calves or something like that, right? But the cross section of the thymus looks like this. It's filled with these different sort of separate chambers. These chambers are separated by sort of a tough fibrous capsule. I kind of think of it like a pomegranate. For those of you who are familiar with pomegranates, you break a pomegranate open, and what do you see inside? You see all the little red seeds, right? And then you see this whitish kind of fibrous membrane that kind of runs through it that kind of separates out these different, regions of the pomegranate, right? So a thymus is kind of like that. But instead of little red seeds, right, these are individual cells that are undergoing things like development into T cells. Here you can tell that there are different regions of the thymic tissue as well. This darker region up here, right? Cells must be doing something there that's different than in this pinker region. We also have these special structures known as Hassel's corpuscles, which, and I love telling students this, to this date, we don't really know what Hassel's corpuscles do, right? I know that sounds like a real lame cop-out, but it just shows you how amazing that there are still things in the human body that we don't fully understand, right? You might argue, well, Hassel's corpuscles, if we have them, they must be really important for T cell development, right? But no, not necessarily, because many other animals, including things like mammals, right, don't even have Hassel's corpuscles, right? So it's still not clear what they really do. But here is a high resolution image showing that you have within this matrix, this kind of spongy matrix of the thymus tissue, right? All of these little round things are potentially developing T cells. So here's another illustration of that. Here's the cross section of the thymus, or one part of it. So you have these chambers that are separated by this fibrous tissue, and you can tell that there are different regions within that tissue, right? This darker region up here is what we call the cortical region, or the cortex. This is where brand new cells that have come from your bone marrow and have left your bone marrow, but are still undecided as to what they're supposed to become. Am I supposed to be a natural killer cell? Am I supposed to be, like a T cell? I don't know, right? When those cells make their way into the thymus, this is where they kind of start their journey. So when they first come in, right away, they have an opportunity to communicate with these orange cells here, right? So these orange cells are different epithelial cells in the cortex of the thymus. Right? So right away, these cells start sending messages to these cells, to the little blue pre-lymphocytes that are coming in, basically kind of trying to convince them, hey, you should try to start making a T cell receptor, because I bet you would make a great T cell, right? So the VDJ recombination starts happening in this region up here within these blue lymphocytes that at this point we call thymocytes, right? Because they were first identified as being, oh, there are these special cells inside the thymus. What are they? So they just named them thymocytes, right? But now we know, right? These are these pre-lymphoid cells that came from the bone marrow and found their way in here, at which point they are being encouraged to try to become T cells. As they start doing things like making T cell receptor, if they are successful at it, they migrate down to the medullary region, right, where they then interact with other types of epithelial cells. So these larger sort of reddish ones here that help to train or take care of actually doing quality control and seeing what types of T cell receptors are being made by these thymocytes. Right? And then ultimately, this is where the developing thymocytes that have made T cell receptor on them, this is where they're told to be either a CD8 positive cytotoxic killer T cell versus a CD4 positive helper T cell, right? And this is where they're going to undergo their own version of tolerance training, right? Okay, last slide for today. I mentioned that the thymus shrinks dramatically as we age, right? So as a result, the functional T cell producing ability of your thymic tissue is slowly replaced over the years by just fat, right? So if you look at a 3-day-old infant's thymus here, right, and compare it to the thymus of a 70-year-old adult, you can see it's very clear the amount of actual functional T cell generating tissue in a 70-year-old is far less efficient than in an infant. Our thymus is largest during our preteen through teenage years, because again, that's when Mother Nature intended us to really work on getting our strongest immune system up and running as part of our fitness, right? To get ready for being selected as a potential reproductive mate. So what I want to leave you with, because I know we're out of time, is these questions. What does this imply about the longevity or the self-renewal capabilities of T cells? Right? How does this contrast with B cell development? And importantly, how might this affect the strength of immune systems in the elderly? Right? Why is it that we all intuitively know that old people, right, have a harder time with having good, strong immune systems? Right? Why is that? Well, you're looking at that, a big part of that right here. Right? And this has profound implications for things like how older people are hit much harder with brand new types of diseases, things like COVID-19. Right? So I want you to think about that. We'll just end here today, but I want you to think about that, and we can take that up at the start of our next lecture. All right. Okay. So that's it for today. Let me go ahead and stop this.